Understanding the evolutionary processes that lead to speciation and diversification on Earth remains a fundamental challenge in biology. I, as well as a team of undergraduate research students in my lab, have been actively involved in a project trying to overcome this challenge. Plants with similar characteristic features are classified into their own families. The sixth largest flowering plant family, the mint family, known scientifically as Lamiaceae, serves as an excellent model for exploring global patterns of plant diversification, evolutionary radiation, plant conservation, and more. Through investigating the patterns and processes of speciation, we can begin to understand how diversity for all life forms arose on this earth in the course of millions of years. By generating a framework of Lamioid, major questions can be addressed involving the origin and evolution of this hyperdiverse lineage and place it in a broader context of understanding the evolutionary patterns and processes of hyperdiverse flowering plant lineages. After all, the processes of adaptive radiation and speciation led to the building of species diversity in nature. Studying a species' history at a molecular level can be an important tool towards understanding the estimation of rates of speciation and extinction, reconstructing historical diversification scenarios, and linking these to ecological and evolutionary factors such as patterns of climatic and regional variations, structural adaptations, as well as life history traits. Recent scientific advancements have facilitated the reconstruction of robust and comprehensive phylogenetic trees, graphical representation of evolutionary relationships in various organismal groups that provide the essential frameworks for the estimation of rates of speciation and extinction, reconstructing historical diversification, inference of diversification timings, and past distribution ranges, linking these to ecological and geological factors. However, for many hyperdiverse lineages, explaining global patterns of speciation and diversification and connecting them to their complex patterns of adaptive radiation and morphological evolution has been a formidable challenge. Inadequate sampling of study specimens, cryptic species, and uncertainty regarding organismal relationships, as well as limited computational power, have often posed problems when researchers have attempted at studying such extensive lineages with interesting questions on their evolution and diversification, like what makes a species rich? What are the causes for certain lineages being more successful in colonizing and diversification than others? How do chromosomes play a role in lineage diversification and radiation? How do separation and dispersal play a role towards shaping the current distributions of large lineages? Did lineages radiate in their original location after reaching new areas? Our project is the solution to the dire need to study the subfamily on a broader genomic, utilizing all the genetic material within the nucleus of the cell level, with the help of high-throughput next-generation sequencing approaches, incorporating hundreds of nuclear molecular markers and genetic data from the green plant cell organelle chloroplast to resolve the incongruences that have been observed so far from previous studies. The findings from this project will be of broad value to evolutionary biologists, ecologists, and conservation biologists by helping close gaps in our fundamental understanding of how plants diversify across the globe from the time life first originated on Earth 4.5 billion years ago. Information gathered from this project will also improve our knowledge of the comparative biology of Lamioid mints, guide new product discovery, and aid in the development of conservation measures for endangered species. I'm Tilottama Roy, Associate Professor of Biology at Missouri Western State University, and this has been a Griffon University production. Share ideas.